So without further ado, let me move on to read the biography of our speaker this afternoon, our distinguished speaker, distinguished Professor Steve Barnes. Professor Steve Barnes is a distinguished professor in the Department of Medicine at the University of Alabama at Birmingham, UAB, and a renowned expert in metabolomics, analytical chemistry, biochemistry, and pharmacology. He directs the Targeted Metabolomics and Proteomics Laboratory, MPL, and serves as senior scientist in the UAB Nutrition Obesity Research Center and the UAB Comprehensive Cancer Center. He is also the former director of the UAB Center for Nutrients, Gene Interaction, and the associate director of the Purdue UAB Botanical Center for age-related disease. With a distinguished career advancing biomolecular research, Professor Barnes has made significant contributions to understanding disease mechanism influenced by nutrition and drug metabolism. His dedication to education and mentorship, including international collaborations, continues to shape the future of metabolomics and foster interdisciplinary research addressing critical biomedical and environmental challenges. Everyone, please make welcome our distinguished speaker, Professor Steve Barnes, for his presentation. Please try to engage by making use of the emojis to welcome our speaker this afternoon. Over to you, Professor Steve Barnes, for your presentation.
Okay, well, thank you really very much. This is a wonderful opportunity to talk to new friends in the making. I really enjoy talking about the science that I've done. I've had a wonderful time in my career, going from a young student, like some people on this call may be, to going through the entire career process and becoming now a distinguished professor. It's a great journey. So thank you for the opportunity to talk to you all. And as I said, the topic is going to be metabolomics and Africa. And I hope we'll have a really good conversation when I'm done with the various slides. Okay, so let's start at the beginning. As I said, I came into this at, I guess, about 20 years old. And so I can tell you a little bit about the beginnings for me and then move into the nature of metabolomics as we know it today. So it all began when first of all, as an undergraduate, I went into I was really heading towards industry, but I fortunately took a 1 year part of the undergraduate degree was actually in industry, and I got to go into the lab of a man called Tony James. And Tony James was the inventor, along with A.J.P. Martin, of gas chromatography. And I very quickly discovered that what I was doing in his lab was the best thing that I ever had done in my life up to that point. And very quickly, I ended up in his office asking what I do next. And so he said, well, you really need to do a PhD. So I had no idea what a PhD was at age 20, but now I was quickly finding out. So from there, I went and applied to graduate school and ended up at Imperial College in London, working for Sir Ernst Boris Chain. And he was the 1945 Nobel Laureate, who was the part of the Fleming and Florey awardees. The three of them got the Nobel Prize. And he was the biochemist who chemically characterized penicillin. And he also, about 15 years later, led to the development of the oral penicillins. But he was also renowned for his work on microanalysis, which was the key to his work on penicillin. And so when I arrived in his lab, they were doing studies using carbon-14 labeled glucose, radioactive glucose, and they were separating, giving it to cell preparations and also isolated organ preparations. And they were recovering the metabolites from it and separating these metabolites out by paper chromatography. That was the technology of the time. And it was the paper chromatography was run in two directions. So you got a 2D separation of all of the intermediates. The intermediates are glycolysis, the Krebs cycle, and amino acids. And once the work had been done and the paper was dried out, it was placed on this frame that you can see on the right. And a Geiger counter was brought up close to the surface of the paper, and it was mounted on a typewriter. And so the radioactivity in that small spot right in front of the Geiger counter was measured and recorded. And then the typewriter moved over one letter and then another letter. And when it got to the end of the line, it dropped down a line, just like old typewriters do. If any of you have seen an old typewriter, these days we really don't do that anymore. But anyway, it meant that with time, the whole space of that chromatogram could be digitized. And so the radioactivity in each frame could be digitized, and then that data was taken to a computer, and from that you could now generate figures and do other analyses. So honestly, this was metabolomics. The actual term metabolome didn't actually come along until 1998. The study of metabolism, I don't know the real start point, but the one that I think of is the Warburg discovery of the fact that the tumor cell was behaving as if it was fermentive. And there's been many, obviously, people that we've read about in books like Hans Krebs and others. I had the great fortune of introducing Hans Krebs at a seminar at Imperial College whilst I was a grad student. So my connection to the past. And I also worked with Keith Mansford. Keith Mansford was the scientific director for Beecham's Pharmaceuticals and was the person who worked with Professor Chain, the work that led to the discovery of the oral forms of the penicillins. And this is me. Aged, I guess, about 22, 23. And I liked building equipment. And this was the equipment I built for doing my metabolomics. And so what I brought was the gas chromatography capability that the department didn't have at that point, what I'd learned with Tony James. And so this is, let me see if I can use something. Oh, here it is. Yeah. Let's use a laser pointer. Yes, you can see it. So this is the chromatography, the oven part of the gas chromatograph. And what comes out of the column goes first of all into, or part goes into an FID detector, which detects the presence of individual compounds. And the other half of it went down this tube into this scintillation cell. So the two photomultipliers here looking at the scintillation events, the radioactivity that was coming through. And then all of this, of course, was digitized, and here's the paper tape recording. Initially, I had to send the paper tape recording via a terminal to the University of London computer, but eventually I got my own computer, standalone. It was about as big as a large refrigerator, and it had 8K of memory. So very, very minor compared to what we have today. Even our phones have gigabytes of capability. Anyway, so I used that to study glucose metabolism in a slime mold that was of great interest, and it was also applied to the work that was being done in the isolated perfused hearts and livers. So that's where it started for me. I also, and I won't dwell on this, but I also got involved when I got to UAB using superconducting magnets. I think I show some pictures of that later. And it meant that I had a knowledge to a degree in NMR metabolomics. And because of that, the dual training that I had from the past in LCMS and NMR, the NIH decided to award me the role of training the next generation. And so we put on 6 national workshops at UAB. They lasted a week, and we trained in total just under 200 people, mostly students and postdocs, but sometimes early emerging faculty. We had a couple of core directors from other universities, and we had one NIH director who came down and got the training. There's a fellow in this picture who is very, very well known in the world of metabolomics, and it's this guy here, and that's David Wishart from the University of Alberta in Canada. And he was a real pioneer in both utilization of NMR and mass spectrometry, but also in the training of people. He ran many courses long before I started mine, and I brought him here in 2015 to do a critical assessment of how well we were doing in our workshop. Anyway, so that was an interesting development period. And the following year, in 2015, what I did was to take the elements that I had put together in the workshop using not just my capabilities, but capabilities of other people who I used to be the teachers in the workshop. I put together a graduate program or graduate course in metabolomics, and I've been teaching that ever since. And we'll talk a little more about that later on in today's talk. But it came to the attention of Daryl Hurt, who was at the National Institute of Allergy and Infectious Diseases. And he asked me in 2016 if I could additionally teach and organize a course in metabolomics to students in Africa. And the students in Africa were in Mali, and NIH had a program with them to try and train people to locally in Africa to be able to do studies on some of the disease outbreaks. The people in Mali were particularly interested in malaria. Anyway, so it was a bit of a challenge, but we put together a program. It was 5 weeks of courses. We held them for 2 hours each day from 9 o'clock to 11 o'clock in the US, and I guess 3 o'clock or 5 o'clock in Africa. We did this by WebEx, and I was able to coordinate with the people locally, Wele Mamadou and Oa Koligbali. I'm sorry if I ruined the names, but it was fun. And so these are 9, I think 8 of the students of the 10 students who took the course, and it was real fun to work with them. So Oa is, I think, still a consultant coordinator in bioinformatics in Mali. And I noticed a recent paper that she had published on the genome-wide SNP analysis of Plasmodium falciparum, showing differential resistance to drug resistance associated loci in malaria transition. So I'm glad to see that she's continuing. She had received training in the US, and therefore was a really good way of being able to coordinate between the students and myself. It was not, it was, there were challenges in teaching the course. Anything at a distance is difficult because of the time zones. Things have got a lot easier. I mean, one of the ways COVID did was to promote the use of things like Zoom. So Zoom has become very, very easy to use, and it's available to everybody. So we had to overcome that. The second problem was that the first language of the Mali students was French, and therefore English sometimes there were little nuances that we didn't understand until we laid out some questions for them. So the classic multiple choice questioning format proved to be actually too subtle. And so we switched to doing problem-oriented problems, which they much more enjoyed because there was outcomes and it required integration of all of their knowledge. and they learned to put together PowerPoint presentations so that they could use this skill set to deliver this message to all their colleagues. The other thing that happened, and I hope we've now more and more got over this, I was told by the NIAID that the students wouldn't ask many questions, that they were intimidated by professors. And after the first week of running this, they commented, students have asked more questions of you in this 1 week than the entire course that we just did, the previous course we just did. So I'm hoping that anyone has a question will want to answer it, and I do my darndest to reply. There were some local issues. Sometimes the power would go out, usually for just a few minutes. We had one day when it didn't come back on. The more subtle ones, we were trying to do some work together on utilizing a MetabHole, and we were uploading some .csv files. And what we didn't realize was, I didn't realize, was that in England and America, the way you represent a number, a complex number, is say 1, 2, 3, period 2, 3, 4. And in the French and the European way of doing things, that period was a comma. And so since we were using a comma separated value format, there were too many commas, and therefore the program would not work. It took about 24 hours before we got a solution to it, and then the students locally were able to get a version of Excel that allowed them to output data into the format that was acceptable for MetabHole.